Hi, I'm Jake, a PhD student from Hidalgo Lab. For my project, I'm researching how experience changes neurons and the circuits they form. When a neuron fires particularly strongly, it can respond by changing shape and growing, making more contacts with its synaptic partners, in turn making it more likely to fire in the future. This could be a fundamental mechanism by which the brain adapts to change. Day to day, I study this in the fruit fly larva, the maggot, because it has neurons with large synaptic terminals connecting to the muscle. Here, these terminals at the tip of neuronal axons branch out like a tree and make multiple synaptic contacts with the muscle. These axonal terminals are plastic, and the number of branches and synapses can change with larval activity, such as crawling. I'm researching genes that could be involved in this process, aiming to understand how the proteins they encode work to regulate plastic changes in the synapses. The findings could be important for understanding how brains work, including the human brain. So, we need to have tools to study our proteins of interest. One that I'm working on today is a protein fusion. It's a copy of one of our proteins that I've genetically modified to be fused to a fluorescent protein. The idea is that when we look at the fly's brains under the microscope, we should be able to see where our proteins are because they'll be fluorescent. They'll glow in the dark, and that's really cool. We have a few fly lines that might have the protein fusion we're looking for, and today what I'm doing is setting up a PCR to find which line is the right one. PCR amplifies a specific sequence of DNA and can be used to tell us whether or not a sample has that specific sequence in it. Today, I'm looking for the gene that encodes my fluorescent protein in DNA I've extracted from flies. The fluorescent protein originally comes from jellyfish, so it shouldn't be in the flies normally. To see the result of the PCR, we run it on an agarose gel and blast it with UV light. If one of my fly lines has the protein fusion, we'll see a band on the gel.
Another project we work in the lab is on how neurons and glia interact, and in particular in the context of regeneration. Lizzie Connolly in the lab is doing her project on this, but today she couldn't come because she's about to submit her PhD thesis. So in normal development, glia help neurons find their way when they establish neural circuits, and then they also wrap around the axons in neurons to insulate them and help them function. But when there is an injury, glia can proliferate and regenerate themselves. And what we're investigating now is whether glia might also become neural stem cells after injury to then make new neurons to re-establish neural circuits and in this way help regeneration.
Hello, I'm Maria. I'm biochemist and I work with protein. Do you know that the protein are one of the most important classes of molecule for life? I combine biochemical and biological tools to understand the role of a protein and how it interacts with other proteins. All to understand whether we can encourage cells to regenerate the damaged nervous system. Now I'm doing a gel and I'm going to check my protein expression. Do you want to see? Come on. This is the gel of my last purification. Do you see the band? Oh, this is one of our protein. It's amazing, eh? It's super nice. We work with E. coli and S2 cell to express protein. Now I'm going to show you the cell culture. Oh my God, the cells are contaminated! No!
Drosophila melanogaster, or commonly known as fruit fly, is one of the most valuable monoorganisms in biological research. They have been extensively investigated for over 100 years and have led to 6 Nobel Prizes. These groundbreaking discoveries include that genes, which are the basis of inheritance, are located on chromosomes, the finding that X-ray causes mutations, the genetic basis of the body plan, the first neural circuits to be worked out, the molecular basis of immunity, and most recently, the molecular mechanism of biological clocks. You might be wondering how such a tiny invertebrate can help us understand human biology. Well, there are 4 reasons why we have chosen Drosophila as a model organism. Fruit flies and us share many similarities. About 60% of genes in fruit flies are conserved in humans, and 75% of human disease genes have functional Drosophila orthologs. Because of this deep homology, flies can help us understand what goes wrong in human diseases and search for possible cures. Furthermore, cells are similar across all animals, and neurons are very similar across all animal brain types. Like humans, flies also have complex behaviors driven by a tiny but sophisticated brain. This is possible because neurons work in the same way. They send electrical signals through neurotransmitters that are conserved between humans and flies. Common neurotransmitters found in humans, such as dopamine and serotonin, modulate different behaviors in flies. Like humans, flies can also learn and form memories and maintain the 24-hour circadian rhythm. They sleep at nighttime and even have a daily siesta. Flies run around, mate, eat, and also sleep, make decisions, learn, remember the past, and predict the future to ensure their survival, just like we do. A refreshing drink for humans, like coffee, would also keep flies awake. If fruit flies drink alcohol, they get drunk, and when they feel rejected, they drink too. Like us, aging in fruit flies also leads to sleep loss, memory decline, and locomotion impairment. Importantly, for work in the lab, fruit flies have a short lifespan and can be bred in large populations, making findings robust. Looking after fruit flies is relatively cheap, and working with them does not raise ethical concerns. The most important reason for using the fruit fly Drosophila in research is its powerful genetics. Like us, Drosophila is also diploid, and each fruit fly inherits 1 chromosome set from the mother and 1 from the father. But instead of the 23 pairs of chromosomes in humans, Drosophila only has 4 pairs of chromosomes, which makes genetic research easier. For each chromosome, we also have balancer chromosomes that are used to prevent exchange between homologs, enabling scientists to maintain families of flies for many years. In this way, some have been kept already for 100 years. Balancer chromosomes have dominant markers that we can see under a microscope in the living fly, like curly wings instead of straight wings, so that genes that we are investigating can be tracked across generations in time and through large populations. Using Drosophila, we can remove genes or have them work in excess to make extra proteins, and then investigate what they do, the consequences to the cells and the brain. The microscopes allow us to see the 2 cell types in the brain, neurons and glia, so that we can investigate how genetic changes modify neuronal and glial shape, connections, and circuits. With cutting-edge genetics tools like optical neuroscience and optogenetics, we can control neuronal activity with light, visualize working neurons with microscopy, and investigate how neural circuits deliver behavior. So, the fruit fly Drosophila allows us to investigate from genes, the proteins they encode, to cells, neural circuits, and behavior.
Hola! Hi! I'm Marta, and I'm the research technician from the Hidalgo Lab. These are the bottles that we normally have the flies on, so you can see that I have here some flies flying around, and some larvae, some pupae, and here is the fly food. So they feed on this, and then we keep them here so that they are ready for when we need to do our crosses. And then when we need to do crosses and we need to select flies, we have to put them in the pad. This pad is connected to CO2, and it makes them fall asleep so that we can actually sort them, like separate the males from the females and the virgins. The male normally is slightly smaller than the female, and it has a dark abdomen, and if you look to the tip of the abdomen, it has like a plate that is the genitalia, while the female doesn't have this and is slightly bigger in size. And then when I want to separate the females from virgins from non-virgins, what I do is just checking the abdomen. Normally the virgins have a very like puffy and wide abdomen, and they normally have a greenish dark dot on their abdomen. That is the meconium. That is basically the last meal that they have before they go into the pupae. When we have to cross a female with a male, we need to know that that female didn't mate with any other male before that. So we need to have them virgins to cross with a specific male with a specific genotype that we want. So in that way, knowing the genotype of the female and knowing the genotype of the male, then we can know exactly what's the genotype of the progeny. So that's really important because it's the first step of all the genetics protocols. So if we don't know, if we can't track the genotype, that's it. We can't actually do the genetics properly.
Oh, hello, I'm Dipanshu, a PhD student, and I'm studying how pathogens interact with our brains. You may have heard of the zombie fungus, which controls the behavior of their host as a mean to protect their own survival. In fact, both host and pathogens compete with each other for survival. Pathogens have evolved different strategies to infect and manipulate their host, and I want to understand how the fungi affect the brain. Do they affect the brain structure or not? Or do they affect how the fly behave? To understand how fungi affect the brain, we need to have a clear view of the fly brain. But how do we do that? Because they are so tiny. The average size of the fly is 3 millimeters. So to dissect the fly brain, I use a stereo microscope and a razor-sharp forceps. This is a time-dependent experiment in which I aim to dissect at least 10 brains in 20 minutes. As soon as I dissect the brain, I have to transfer the brain into the fixative to stop all the reactions in the brain and to make the brain ready for the next step, that is the immunostaining.
Hi, my name is Nasser Shamsi, and I'm a PhD student at the Hidalgo Lab. In my project, I am trying to understand how neural circuits are formed in development. Interesting fact, there are 86 billion neurons in the human brain that come together to form functional circuits, and I am trying to understand how neural circuits are formed, how do neurons establish connections in the brain to form the final functional brain, and how do they know which other neurons to connect to. I am studying whether a gene is involved in neural circuit development. This gene makes a protein. To see which neurons in the brain that have this protein, I have tagged it with GFP, a fluorescent molecule, and I'm using an antibody to detect this GFP. This antibody is fluorescent, and I will look at it under the microscope.
Hi, I'm Gui. I'm a postdoc from Hidalgo Lab, University of Birmingham. So I study how the structure of central nervous system changes responding to the environmental stimuli. That's what we call structural plasticity. So you may have come across the brain plasticity before. For example, the people who are good at playing the musical instruments, their brain develops differences compared to the people who don't play. So in the nervous system Drosophila fruit flies, it is also plastic. For example, when keeping the flies in constant light or darkness, the size of the brain can be altered, and also the synapse number and also the cell shape. So what I want to understand is how the neurons in the brain are plastic and how the synapses can be changed responding to the environmental stimuli or experience, and also how the genes are involved to enable the brains to be plastic. Hello everyone, I'm at the confocal right now. So I believe that Nasser has already explained to you how the immunostaining works. So now I'm gonna introduce you to the confocal and how to visualize the staining at the confocal. So this is the slide with the brains that we have already mounted. You can see, maybe you cannot because they're really tiny like dots, but the microscope can enable us to see the brains clearly. So we're gonna move this back. So this is confocal. The confocal works with lasers. The very cool thing I found about confocal is that it can focus on really fine focal planes. Like it can scan the samples by microns, and that each micron is in focus and not affected by other microns. So if you can come closer. So this is the scanning I just did, and you can find that you can see different parts of the brain clearly. For example, this is the mushroom body, and this is the antenna lobe, and this is the optic lobe. And here are the neurons from the mushroom body. So you can see that different parts of the brain, they can be scanned really clearly and in focus.
Hi, so this is our behavior room where we do all of our behavior experiments. Here I'm going to show you a very cool, a simple and cost-effective assay. It is called negative geotaxis assay, or in simple terms, the climbing assay. For this assay, all you need to do is put some flies into these vials. Different labs, they use this assay to show that flies can have Parkinson's as they don't climb. So once we transfer our flies to these vials, we tap the vials and count the number of flies which climbed the 2 centimeter mark. The idea is that the young flies, they are more active as compared to the old flies and climb faster. We record these flies and can also use different softwares to analyze the recording. So yeah, that's about the climbing assay.
Hello, welcome to the activity part. Today we're going to make a fly brain by ourselves. So Drosophila adult brain functions similarly to human brains. They're both modular, and different modules have different functions. So to make the fly brain, we simply need papers in different colors, pencil and glue. So now let's get started. So the first thing we need is a brain template. So this is the shape of an adult brain. And then let's go from the outside into the deeper inside. So the first thing is the visual system of a fly, which is the optic lobe. This is like our eyes, which is involved in the visions. And then let's go to the olfactory system. So it's like our nose, which can help the flies to smell the environment, to help them to see where they can get the fruits. And this is called antenna lobe. Okay, and now let's get to the deeper inside of the brain, which is the central complex. And the central complex is like a controlling center of the Drosophila, which is composed of 3 parts mainly. The first part is mushroom body, which is mainly involved in regulating learning and memory, like our hippocampus. There are 2 mushroom bodies in the Drosophila brain, one on the left and one on the right. And then the second part is the ellipsoid body, which is like a motion center, so it can control the flies for the body coordination or the avoidance or the flying, walking motion functions. And then finally is the fan-shaped body, which is also involved in regulating the motion functions. So the fan-shaped body is on top of the ellipsoid body. So that's it. This is like a simple version of the fly brain, which is composed of the main functional parts of the brain.